Yeah, recover. Come on. Okay. Okay, so we are going to talk about the neuromuscular junction. So we are going to talk about the neuromuscular junction. So what is a neuromuscular junction? So a neuromuscular junction, I can say it's a junction between the nerve and the muscle. Okay, it's a junction between the nerve and the muscle. As we noted out last time, we talked about the action potential, how the action potential are being generated. We talked about that, and the videos are still there on Telegram. We mentioned that the action potential is produced by the action of sodium channels, sodium voted gated channel and potassium gated voted channels. We mentioned that there is always a resting membrane potential for any particular tissue. We talked about the nervous tissue having the resting membrane potential of about could be negative 30 something. You should no negative 90 somewhere, negative 90 somewhere there, or negative 90 or negative 7 somewhere. That's what we talked about last time. That's what we said last time. And then from there, we mentioned that this resting membrane potential is determined by the potassium concentration, because it's the one that's where the value closes to. And we discussed that, we noted that, we talked about whatever that makes the potassium being the one that is responsible for determination of the resting membrane potential. We said that after that, whenever there is a stimulus, when you stimulate the nervous tissue, we mentioned that the nerve becomes depolarized due to sodium influx. Well, we talked about that. The whole video is still there. Now, I don't know if it can make sense if we can explain this on just one slide, because like the whole slide has got everything, and then I would add up the other things next time when we meet. So the junction between the terminal branch of the nerve fiber, this is the nerve fiber. So this nerve has got the board which is there, the cell board which is there, then it has got what we call the axon terminal. The axon terminal, the ones that are attaching on the muscle fiber there. And I can tell you that one, this motor neuron A, it can be called a motor unit. What do they mean when they say a motor unit? That one is a definition. It comes in histology, in physiology itself. They can ask you what is a motor unit. A motor unit is a nerve, and then which innervates more than one muscles. So these muscle fibers are innervated by that nerve, by that neuron, and that is, it is called a motor unit. Okay, neuromuscular physiology. So what is the neuromuscular physiology? We've talked about the neuromuscular junction to be, that's the junction between the nerve terminal and the muscle where it binds. So let me summarize it somewhere. Or sometimes they call it a synapse. A synapse is the synapse is where the two nerves meet, or when a nerve and the muscle meet, it's called the synapse. So a neuromuscular junction or a myo is called the muscle. Neuro, so myoneural junction. So it is not just neuromuscular, but it is also called myoneural junction. Is a chemical synapse formed by the contact between the motor neuron and the muscle fiber. The motor neuron and the muscle fiber, we've talked about that one. It is at the neuromuscular junction that a motor neuron is able to transmit a signal to the muscle fiber, causing the muscle contraction. So in short, the nerve that carries the action potential, that will lead to the muscle contraction. So that connection that is there, the synapse that is there, it is called the neuromuscular junction. So if you've got the myelinated nerve fiber, the motor neuron, we know that the motor neuron these are neurons that bring about an impact or an effect, contraction, for example. We know that. So having talking about that, we quite know that the action potential is generated from the cell board, and then we mentioned that the cell board, where the action potential will move along it starts from the axon hillock. We mentioned that. That's why it starts from. When it starts from there, it will be propagated or conducted along the axon terminal, along the axon, and then from the axon there, it will go to axon terminal. When it reaches the axon terminal there, it will lead to what to call the calcium influx. It will lead to what to call calcium influx. Calcium will enter a lot in that terminal button. When a lot of calcium enters the terminal button, what happens is that it triggers the release of neurotransmitters, for example, acetylcholine. So when that acetylcholine has been released, it is the one that will move from that axon terminal to be released by exocytosis. It will go into the cleft, also called the synapse. It will go into the synapse. When it gets released there by exocytosis, it will move and then to bind on the muscle membrane, which is also called post-synaptic membrane, because it is after the synapse. The one that is before the synapse is called the presynaptic membrane, or presynaptic nerve fiber, or nerve terminal, if you will. So now, when that acetylcholine binds on the muscle fibers, it triggers the opening of channels. For example, sodium channels. So a lot of sodium that is surrounding that area start influxing or entering the muscle, and we know that the sodium, so when sodium enters there, it triggers it will activate what you call, it will depolarize the cell, because sodium is entering in the cell. When sodium enters the cell, it will depolarize the cell. When the cell becomes depolarized, what happens is that it will activate the release of calcium from the tissue bowels. When that calcium gets released from the tissue bowels, it's not that it will move along the tissue bowels and go and activate what we call the troponin, tropomyosin, and the titin complex, leading to what you call muscle contraction. That's the summary of the neuromuscular junction. So I'm going to summarize it tomorrow. I'll talk about it for a while, and then we'll actually proceed to the Krebs cycle, because we did the Krebs cycle, and for this time around it'll be a bit simple and straightforward. Okay, so that summarizes everything about the neuromuscular junction. Now let me tell you something. This is the let me just say the page. This is the page that will summarize everything about the neuromuscular junction. So we are saying that there is an axon. Now we're saying this neuron is a motor neuron, meaning that it brings about an effect from the central nervous system, from the spinal cord or the brain direct, into bringing about an effect to the muscle tissue, bringing about the muscle contraction. So now we're saying that nerve terminal, and the muscle, they're forming what you call the synapses. They meet somewhere. Whenever these two things meet, they form what you call synapses. The synapses can be between the nerve and the nerve itself. It can be between an axon and the dendrite, and it is called dendroaxonal synapses. Sometimes it can be on the board, and it is called the axonal soma synapses. So it depends on where it is attaching. But this kind of synapses is called the neuro or myroneural junction or neuromuscular junction, because you have got the muscle and the neural being involved in there. Okay, so now we said that the action potential. Let me just try to annotate something. It's just that I'm having problems with my channel. It's becoming irrelevant. Share something new. Okay. Boom. Okay, here you go. So now we drew the nerve last time, and we said this is the neuron. It has the axon here, and then you've got the axon terminal. On the axon terminal, you've got something like buttons. They are called terminal button or axon terminal just. So in this axon terminal, that's where the neurotransmitter is stored. And then we said we've got the dendrites, and we said these neurons can be classified according to the number of poles that they have, or the functions they play, or indeed the length of the axon. Yeah. So we talked about the Golgi type 1 and Golgi type 2. Remember all those things. Don't forget that. So now we have we said this action potential is generated within the cell and within the body, and it is generated because of what? We said because of calcium, sodium is entering there. And the video is still there on Telegram, just watch it. And then we said that the axon, this one can be myelinated or unmyelinated. The myelinated, we said the action potential jumps from one node of Lambert to the other, node of Lambert to the other, because we said on the nodes of Lambert, we have a lot of sodium channels there, which are called sodium volet-gated channels, such that the action potential jumps from one node of Lambert to the other, meaning that the action potential is conducted fast on the myelinated nerve fiber than on the unmyelinated nerve fiber. Okay, so from there, when that action potential moves, okay, even if it is not jumping, we said the process by which the action potential jumps from one node of Lambert to the other is called saltatory conduction. So now that is on myelinated fibers, but if they are not myelinated, they are called unmyelinated nerve fibers. Now we are saying that when that action potential moves from the cell body that is produced going in this direction, it will go to the axon terminal A. When it reaches the axon terminal there, it will trigger it will cause what you call the opening of channels. When those channels open, what happens is calcium will start influxing, because you have calcium outside the cell. So calcium will start entering the cell itself on the terminal, sorry. So we are going to have an influx of calcium. So we are saying when the action potential reaches this terminal button, it will trigger the opening of channels. When those channels open, what happens is that they will induce the influx of calcium into the terminal button, so that those calcium start influxing. When those calcium enters those terminal button, the calcium is not that to trigger. We said, remember what we talked about last time? We said calcium is important in the release of neurotransmitters. and activating enzymes. So in this case, calcium is a second messenger. What happens is that it binds on those channels that are within the same terminal buttons. Let me enlarge the terminal button. Let me enlarge it. It's like this, very big one, but this is molecular level. So we assume it's under the molecular level. So it will, we said when the action potential reaches this side, it will trigger it will cause the opening of sodium channels. So that sodium, not sodium, sorry, excuse me, it's calcium. So it will trigger the influx of calcium here, calcium ions. And then remember what we said, in the body here or the nerve, there's a production of a lot of things because we've got mitochondria, we've got all those sorts of things that side. So they will produce neurotransmitters. Neurotransmitter will be stored in the terminal buttons. That's what we said last time. And they move out to go by anterograde and retrograde. Remember what we said last time? So in here, you're going to have vesicles containing neurotransmitters like acetylcholine. Okay. So what happens is that when there's that influx of that influx of calcium in here, it will trigger these membranes, these vesicles that have neurotransmitters. They'll start like fusing with the membrane. They'll start moving towards this side because of the influx of calcium in the terminal button. And then this neurotransmitter will be released by what you call exocytosis. They'll be released by exocytosis, and then they'll come here. So you've released acetylcholine here, acetylcholine, ACH. So when it is acetylcholine in here, you have got a muscle this side, the muscle this side. You've got the muscle this side. So let's assume this is our mass. So now this mass of ours, this form, now this region between the nerve terminal and the muscle is called the junction, which is called the neuromuscular junction or neurojunction. Okay. So now when that acetylcholine has been released into the cleft, it is called cleft. Synaptic cleft. So this one before it is called presynaptic. Well, this one is called postsynaptic membrane. This one is presynaptic neuron. So that when the neurotransmitter is released there, it will go into the interstitial space here, or let me just say into the cleft. When it reaches there, it has got receptors on the muscle fibers here on the postsynaptic membrane. It binds there. When it binds there, it triggers the opening of those channels that it bound on. So they will open up. They'll open up. When those channels open, they will lead to the influx of sodium, because we said sodium is out, it's more on the outside of the cell. So sodium is a lot in here. So what happens is that sodium will start influxing. Sodium, oh my God. Sodium will start influxing to start entering in this direction. Let me just change the color. Sodium will start influxing in this direction. So when sodium enters in there, remember what happens when sodium enters the cell? It depolarizes the cell, meaning that the cell becomes so excitable. It becomes so excitable. So you're going to have this cell being depolarized, the muscle cell being depolarized. When it gets depolarized, what happens is that it will trigger the release of calcium somewhere in the tissue bowels along the same muscle fibrils or the muscle fibers. So they will release what you call the calcium. In short, the action potential will trigger the release of calcium within the same muscle fibrils. And then that calcium is involved in the muscle contraction, where we talk about tropomyosin, troponin, and titin, and also other substances that we talk about concerning the muscle contraction. So I want you to know that that is all about the neuromuscular junction. And then there are conditions like myasthenia gravis. In myasthenia gravis, what happens is that it's an autoimmune disease whereby the body attacks these receptors for neurotransmitters like acetylcholine. They attack them. When they attack them, they destroy those neurotransmitters, such that when acetylcholine gets released, it will bind here, of course. It will bind here. It won't bind here because those channels have been destroyed. Let me just go direct to a good presentation that will make sense, because that's what is summarizing everything that I've talked about. Oh my God. Okay, so it's a summary, by the way. It's not something that is very complex, something that is very simple and straightforward to understand, unless if you want to take it personal. Okay, you share. So that has summarized everything. I've talked about the application. So this is the neuromuscular junction. So number one, the action potential will reach here. So the transmitter synthesized and then they stored in the vesicle. I talked about that. Number two, action potential invades the presynaptic terminal. When the action potential enters here, when it reaches here, it depresses the presynaptic terminal, opening the channels. Which channels? The calcium voltage or voltage gated calcium channels. Calcium, number 4, calcium will start entering. When that calcium enters there, calcium causes vesicles to fuse with the presynaptic membrane. I talked about that, releasing the neurotransmitter into the cleft there. So the transmitter is released into a synaptic cleft by excitosis. I talked about that. Number 7, the transmitter binds to the receptor molecule in the postsynaptic membrane. I talked about that one. That will lead to opening of postsynaptic channels like sodium gated, voltage gated sodium channels. So postsynaptic current causes excitatory or inhibitory postsynaptic current. That is okay. So it can be excitatory or inhibitory, but in this case, when you want to contact the muscle, it's excitatory. That's what happens. So it will trigger it will cause the same thing that we've talked about. So now what happens is that acetylcholine that was raised, it is supposed to be degraded, it is supposed to be destroyed by what we call acetylcholinesterase. So acetylcholinesterase is an enzyme that breaks down the acetylcholine. Acetylcholine into acetylcholine and choline. And we'll actually talk about that when we meet. Okay. So I don't know if tomorrow we can do physiology or we do biochemistry, but since Madam Banda is not there, we can do biochemistry. This one we'll do it again. It's just an introduction to you so that you understand it. Now, under myasthenia gravis, what happens is that these receptors that are on the postsynaptic membrane are destroyed by the body cells. They recognize them as non-self antigens or non-self proteins. Therefore, they attack them and destroy them, such that when acetylcholine comes here, it won't bind here, and that is called myasthenia gravis. So there are drugs that we give in myasthenia gravis, and I'm going to tell you this the next time we'll talk about it. So this summarizes everything that we've talked about. All these slides are just talking about the neuromuscular junction, and I made them very simple and straightforward so that people can understand them properly. So that's all for today. I've talked about this. Everything is there. You can explain these things. You can explain. So somebody will summarize this for me when we talk about physiology. We'll meet, I think, on Monday for physiology. Okay. It was nice talking to myself this evening. It was nice. Okay. Yeah, but I understand people are tired because they were writing the pathology test. Yeah, awesome. So now, next time we'll meet to talk about other things. We can do the revision of the same things. Yeah, but I've summarized everything about the neuromuscular junction, and you need to know it. The way I've explained it is the way I'm supposed to explain it. Maybe I'll just talk about the formation of the acetylcholine, how it is formed by the enzymes and everything, and the myasthenia gravis, because it is highly yield. Okay, thank you so much, you people. Have a blessed night, and God bless you. William M. Chance. Thank you.